Hello, welcome to the GMP Guide, your best resource for pharma knowledge. By the end of this video, you will learn all the basics you need to know for your work and gain the confidence to ace your next job interview in the pharmaceutical industry. Quality by design, QBD, quality must be built into the process, not just tested at the end. If it isn't documented, it didn't happen. Documentation is the absolute proof of compliance and process execution. Data integrity. All records must meet the ALCOA plus criteria: attributable, legible, contemporaneous, original, accurate, plus complete, consistent, enduring, and available. Validation is proof. Validation, e.g., equipment, processes, cleaning, is the documented evidence that a system consistently achieves its intended purpose. Control of change, change control. Any change that could affect product quality must be formally reviewed, approved, and documented before execution. Risk management. Decisions about quality should be based on a scientific assessment of risk to the patient and product. Right first time. The philosophy of aiming for zero errors and deviations to minimize waste and risk. Training is mandatory. Every person must be trained and qualified for the tasks they perform. 2 signatures for critical steps. Critical manufacturing or testing steps must be witnessed and verified by a second qualified person. Segregation of duties. No single person should control all steps of a critical process, e.g., manufacturing and release testing. Management responsibility. Senior management is ultimately responsible for providing the resources and infrastructure to maintain a compliant quality system. Cleanliness prevents contamination. Maintaining a clean and orderly environment is fundamental to preventing microbial, particulate, and cross-contamination. Batch concept. All processes relate to a defined batch size, starting materials, equipment, and defined endpoints. Process ownership. The manufacturing team owns the process. Quality assurance, QA, owns the compliance. Preventative maintenance, PM. Equipment must be regularly maintained and calibrated before it fails. Calibration is key. All measuring instruments must be calibrated against a traceable standard. Deviation management. All unexpected events or departures from the procedure must be immediately documented, investigated, and root cause identified. Corrective and preventive actions, CAPA, after a significant issue, define actions to correct the immediate problem, corrective, and prevent recurrence, preventive. Standard operating procedures, SOPs. All critical activities must be performed according to approved, written instructions. Reconciliation. All materials, starting materials, intermediate product, labels, bulk product, must be accounted for at the end of a process. No dry labeling. Never record data on scratch paper or memory. Record data directly onto the official document. Contemporaneous. Traceability. Every component, person, piece of equipment, and action related to a batch must be fully traceable. Label control. Labels are critical. They must be stored securely, issued carefully, and reconciled completely to prevent mix-ups. Good documentation practices, GDP, use indelible, non-smearing ink and follow the procedure for corrections. Single line out, initial, date, reason. Equipment status. Clearly label all equipment with its current status, e.g., clean, dirty, maintenance, quarantine. Personnel health. Sick personnel, or those with open wounds, must not work in critical production areas. Gowning is a barrier. Gowning procedures create a barrier between the operator and the product, protecting the product from contamination. Pest control. The facility must have a documented, routine, and effective pest control program. Retention samples. A reserve sample of every released batch must be kept for a period after the expiration date. Annual product review, APR. Batches must be reviewed periodically, usually annually, to spot trends and ensure continued process capability. Out of specification, OOS. Any test result that falls outside the acceptance criteria triggers a mandatory, documented investigation. Quarantine is mandatory. All incoming raw materials and finished products must be placed in a quarantine status until formally released by QA. Temperature and humidity control. Environmental conditions in storage and manufacturing must be monitored and controlled to protect material stability. Regulatory inspection readiness. Assume regulators, e.g., FDA, EMA, could arrive at any time. Always be prepared. 2. GXP-specific and data integrity principles, 36 to 70. This section focuses on laboratory, clinical, and electronic record standards. Good laboratory practice, GLP, test facility management. Clearly defined organizational structure and responsibilities within the lab. Reference standards control. All standards used for testing must be properly sourced, characterized, stored, and periodically re-verified. Raw data integrity. The original observations, calculations, and printouts must be maintained and archived. Archiving. Test records, specimens, and reports must be securely archived for the required retention period. Method validation. All analytical methods used for testing must be proven to be accurate, precise, specific, and robust. Equipment logbooks. Every piece of lab equipment must have a logbook detailing its use, maintenance, and calibration. Non-clinical safety. GLP primarily focuses on the safety and efficacy of investigational products during non-clinical, animal, studies. Sponsor responsibility. The sponsor, company, has overall responsibility for ensuring the study is conducted according to the GLP regulations. Test system integrity. For animal studies, the test subjects must be identified, characterized, and housed properly. Reagent dating. All reagents and solutions must be labeled with their preparation date, concentration, and expiration date. Good clinical practice, GCP, informed consent, IC. A voluntary process where the subject understands the study's risks and benefits before agreeing to participate. Institutional Review Board, IRB, approval. No clinical trial can begin without the review and approval of an independent ethics committee, IRB, IEC. Clinical trial protocol. The detailed plan for the study. It must be followed strictly, and deviations must be documented. Subject welfare. The rights, safety, and well-being of the trial subjects are the most important considerations. Investigators Brochure, IB, a summary of all the clinical and non-clinical data on the investigational product. Source data verification. The monitor verifies that the data recorded in the case report forms, CRFs, matches the subject's original source records. Adverse event, AE, reporting. All untoward medical occurrences must be documented and reported promptly to the sponsor. Serious adverse event, SAE, reporting. SAEs must be reported immediately to the sponsor and, potentially, regulatory authorities and the IRB. Blinding. Procedures to ensure that one or more parties, subjects, investigators, or assessors, are unaware of the treatment assignments. Drug accountability. The investigator must meticulously log the receipt, dispensing, and return of all investigational medicinal product, IMP. Computer system validation, CSV, and data integrity. System lifecycle. Computer systems must be managed through a formal lifecycle. Planning, specification, configuration, verification, and retirement. 21 CFR Part 11. The U.S. regulation governing electronic records and electronic signatures, ERS. Audit trails. Electronic systems used in GXP must have secure, time-stamped audit trails that track all changes to data. Access control. User access must be limited to those with approved roles, and passwords must be secure. Disaster recovery. A plan must be in place to recover data and systems in case of a failure. Backup and restore. Data must be backed up routinely, and the ability to restore the data must be tested periodically. Validation status. A validated system must be maintained in its validated state through change control and periodic reviews. Data migration. Transferring data from an old system to a new one must be formally validated. Security. The system must be protected from unauthorized access, loss, or corruption. Electronic signature equivalence. Electronic signatures must be legally equivalent to handwritten signatures. Metadata is data. The data about the data, e.g., timestamps, user IDs, instrument IDs, is as critical as the primary result. Standalone assessment. Even if a system isn't networked, it still requires validation if it generates GXP data. Off-the-shelf software, COTS, Even commercial software requires validation for its intended use. System retirement, the procedure for decommissioning an old system and retaining the historical data. System owner, a specific individual must be accountable for the ongoing compliance of an electronic system. 3. Material, facility, and release principles, 71 to 100. These principles focus on the physical manufacturing environment and the flow of materials. First in, first out, FIFO. Material should be used in the order it was received or manufactured to manage expiration dates. FEFO, first expired, first out is often preferred. Supplier qualification. All raw material suppliers must be audited and approved before use. Specific sampling. Only approved personnel, following a detailed SOP, can take samples of raw materials. Dedicated equipment. Equipment used for highly allergenic or toxic materials should ideally be dedicated or require rigorous cleaning validation. Aseptic technique. Operators working with sterile products must demonstrate impeccable technique to avoid microbial introduction. Line clearance. A thorough check must be performed between batches to ensure all materials from the previous batch are removed. Positive pressure. In aseptic areas, air should flow from the cleanest zone outward to prevent contaminants from flowing in. In process controls, IPC, checks performed during manufacturing, e.g., weight, hardness, pH, to ensure the process remains within its validated state. Reject, waste documentation. All rejected materials and process waste must be accurately documented and disposed of according to SOPs. Certificate of Analysis, COA. A formal document from the supplier or QA that confirms a material meets its required specification. Water system validation. The purified water system must be validated, monitored, and maintained to prevent biofilm and microbial growth. Sterilization validation. Cycles, e.g., autoclave, dry heat, must be validated to prove they consistently achieve the required sterility assurance level, SAL. Product recall. A formal, documented, and rapid procedure for retrieving a distributed product that poses a health risk. Quality agreement. A contract between two GXP parties, e.g., manufacturer and packager, defining the quality responsibilities of each. Container closure system. The primary packaging, bottle, cap, vial, stopper, must protect the product and not interact negatively with it. Stability testing. Finished product must be periodically tested under controlled conditions to confirm its assigned expiration date. Media fills. Periodic simulation of an aseptic process using a growth medium to prove the process is capable of producing sterile product. Gowning checks. Routine checks must confirm that operators are correctly gowned and protected before entering critical areas. Cleanroom classification. Rooms are classified based on the level of particles and microbes permitted, e.g., ISO 5, Grade A. Holding times. The maximum time a material, intermediate product, or cleaned equipment can be held before further processing. Critical versus non-critical. Understanding which process parameters directly impact product quality and which do not. Final product release. The official action taken by the quality unit to permit the distribution of a finished batch. Cross-contamination prevention. Utilizing separate equipment, airlocks, and dedicated clothing to prevent material from one product from mixing with another. Critical utilities. Systems like water, HVAC, and compressed air treated as critical and require validation and monitoring. Security of records. Paper and electronic records must be stored securely, protected from damage, loss, or unauthorized access. Annual training review. All employees must have their required GXP training reviewed and updated annually. Disinfectant rotation. Different types of disinfectants must be rotated in clean rooms to prevent microbial resistance. Airlock use. Personnel and materials must follow the documented procedure for using airlocks between rooms of different classifications. Labeling accuracy. The content on the label, drug name, strength, batch number, must be 100% accurate before application. Internal audits. Companies must routinely audit their own systems to identify and correct weaknesses before regulators do. If you want to master these principles and stay ahead in the pharma industry, make sure to hit that subscribe button and turn on notifications.
all I wanna be.